And thanks so much, Karina, for a kind introduction and the invitation to speak here today for this audience and to everyone that has come to listen to us. Very nice to talk to this audience, and especially to members, I'm sure, at TMBL, where I did my PhD, as Karina pointed out. Just wanted to say that I did two postdocs, was a single postdoc in labs that moved from one place to the other, but still they were indeed an evolution, so maybe they were two indeed. Okay, so the central goals of my laboratory are to understand how stress responses modulate organismal homeostasis and how can we harness that knowledge for potential therapeutic applications. And because we are interested in studying homeostasis early on, we decided to use mostly a model of extreme homeostasis disruption, which is sepsis, which is a disease that affects nearly 50 million people all over the world every year. And as a result, about 25% of those, unfortunately, will die. And just in 2017, about 11 million people died as a result, and to this day there is no specific treatment for this condition. Now, sepsis might not be known to all of you. This is a systemic inflammatory response syndrome that normally originates resulting from an infection, most often by bacteria, but also and in growing numbers from fungi, and also from a viral origin, which can progress for multi-organ failure. It has been associated for forever, let's say, or was considered an infectious. Always looked from the side of the pathogen, but with introduction of antibiotics, it became clear that even the elimination of the causing agent does not guarantee that the patient is going to survive, and therefore people started to pay attention to actually the host rather than the infection itself. And as a result, a number of attempts to get better outcomes at this condition were done. So there had been dozens and dozens of clinical trials trying to block the initial key mediators of sepsis, most notably by trying to block TNF. And also because this systemic inflammatory response ultimately causes a compensatory response that leads very often to immunosuppression and leaves the patient susceptible to a secondary infection. There's now a school of thought that thinks it's important to do immunostimulation. However, real-world data so far does suggest that neither the block of the key mediators of sepsis and also not immunostimulation seem to be good enough improvements to treat this condition, and therefore, based on our work, which I showed you at least a little part of it, we are proposing that actually maybe there's a third way to be associated with the standard of treatment, and that is the induction of disease tolerance. So what's disease tolerance? I would like to share with you the idea. Some of you are familiar, especially those at IGC, which have heard myself and Miguel Suarez a number of times talking about disease tolerance, but for the ones that have not been exposed to the concept, we argue that a successful immune response depends not only on resistance mechanisms, which are classical type of responses that the host uses to eliminate the inciting pathogen, but also depends on a group of processes, most of them are not really well known, which aim to deal with the consequences of the presence of the pathogen or the resistance mechanisms used by the immune system to deal with the presence of the pathogen, which commonly we can call immunopathology. And so this group of processes that mostly aim to control tissue damage are involved in a significant and key arm of this response, and it's only the synergy of to the function of the organism. Now, in this conceptual framework, we have a number of years ago shown that DNA damage response that could be caused by a low dose of anthracyclines, which are usually used to treat cancer, may cause strong protection against sepsis, and that protection is actually due by the induction of disease tolerance rather than the promotion of resistance to infection. In addition, we have also observed that the lung is actually a very organ where this protection, this disease tolerance is important, and actually initiation in the lung might not only be necessary but actually sufficient to improve this condition, and it can be induced by DNA damage. Now, this is, you can read about it. We published this a number of years ago, but this is to introduce the topic that I wanted to talk to you today, which is about the perturbation of an additional core cellular function that can also lead to protection against sepsis. And this is largely the work of a very talented graduate student in the lab, Enrico Asso, who's now in Vienna. And also with the help mainly from André Barros and Tiago Velho, two additional graduate students in the lab. And this really stems from the initial observation that DNA damage response can be protective. And so we therefore asked whether there would be other perturbations of core cellular functions that could have similar outcomes in the protection against severe infection. And this is the result that we published recently. And actually there's back papers showing that actually inhibition of protein synthesis in the mitochondria can have very important impacts in immune responses, both in induction of disease tolerance and also counteracting autoimmune responses at the level of T cells. But really this started with the question whether there would be additional perturbations in core cellular functions that could be protective. We, as I mentioned, showed that DNA damage can be one of them. So we started this project with a small in vivo screen using drugs that are approved for clinical use and for which they are known to perturb some of these cellular functions. And I should say that we didn't find the phenotypes, strong phenotypes for most of them. We did find phenotypes for drugs that acted on the ribosome, specific on the ribosome, and the ones that we tested with most success are actually the group of tetracyclines, but also several other antibiotics that target the ribosome. And by the way, 50% of all classes of antibiotics act this way, even if they have particular types of mechanisms. So because we wanted to explore then the role of these antibiotics, and we wanted to uncouple resistance from disease tolerance, and of course antibiotics have a direct impact on the pathogen, we then engineered a strain of E. coli that would be resistant to these antibiotics so that we could look for an impact on disease tolerance. And here are the results. I think you can appreciate that when we use the low dose of tetracyclines, here in particular doxycycline, at the time of the procedure, 24 hours and 48 hours after we induce the infection, we had a very substantial improvement in survival, which is also substantiated here in this graph by looking at temperature. I should remind you that mice normally react with hypothermia to a severe infection, and so we can appreciate that mice that were treated with doxycycline have temperatures that are much closer to the normal expected values for a mouse. Now, this was not only the case when we use tetracyclines, which have a particular target site in the mitoribosome, but if we use a totally unrelated antibiotic that also acts on the mitoribosome, that is called F-nicol, for example, we also observe a substantial improvement in survival, and therefore this seems to indicate there's a common mechanism improving disease tolerance and improving survival in the models of sepsis in mouse. As expected, when we look at bacterial burdens in the peripheral blood of these animals, but also in the classical targets, main target burden, which would be expected because we are using a strain that is resistant against we also can look at disease tolerance and see that this is a mechanism largely based on induction of disease tolerance, because if you looked at serological markers of liver, general markers of organ lesion, and renal and muscle, they are on average substantially decreased. Tissue damage going on when we treat animals with these drugs. This can also be demonstrated by histology analysis in the sepsis, again, the liver, the lung, and the kidney. You would agree that doxycycline substantially improves the architecture and the inflammatory infiltrates in all these organs, and there is not only less damage, but also over time they improve considerable with this treatment. Now, we also tried additional models. In addition to a bacterial infection, we also look for a viral infection, for example, with the influenza delivered to the lungs through the nose. And here, while we observe a similar severity in the presence and absence of the, when we actually observe a faster kinetics of recovery as measured by the weight that comes to baseline levels in non-treated animals. In the setting of a parasite infection like with Plasmodium, while these drugs, and this is known for a very long time, and actually used in the clinical to treat observable effect on actually the survival, suggesting that actually these effects are rather specific to bacterial and viral infections and not to parasitic infections, or also in the case of the fungal infections where we saw no advantage in survival in animals that were treated with doxycycline. So trying to figure out what was going on, we initially looked into the lung, because the lung is one of the first organs to fail in the setting of sepsis and is particularly important for the pathophysiology of this condition. First of all, we did demonstrate that we actually could get a substantial improvement by the direct delivery of this drug to the lung, which also suggests a local mechanism that perhaps later becomes systemic, but we could mimic the survival levels by delivering the drug directly to the lung. So we tried to figure out what was with bulk analysis by RNA-seq of lungs from animals that were infected to extract the classical or the infection, and then comparing it to animals that were actually treated with these drugs in the presence or absence of infection. And there were not many genes changed by the presence of in either non-infected or infected conditions. We did find a very robust signature of a few dozen genes that all pointed in the direction of a differentiation response, which is known at the level of the skin in keratinocytes, and so this to us probably means that these drugs are recruiting cells that are involved in repair and differentiation, and so re-establishing key cells for the function of the lung, which we have now tried to go deeper and looking, for example, at the possibility that these drugs are engaging stem cells. Here we only looked for basal cells, which is only one of the types of cells that are present in the lung and give rise to several of the components of the respiratory epithelia. And by using a mouse that where we can deplete basal cells, we did not observe a substantial decrease in the protection by these drugs, suggesting that it's probably acting in additional types of stem cells in the lung, not only basal cells, but the other types of cells. And so this is something that we are still exploring, but it's clear that we not only observe a decreased damage of the lung, but we also observe a faster recovery in the recovery of the epithelia in the lung. So this is still under investigation. we actually decided to turn to the other key organ in sepsis, which is the liver. And very early on, at 8 hours post-infection, contrary to the one in the liver in infected or non-infected conditions. And this made us turn to another unbiased method to potentially at causative mechanisms, and therefore we did unbiased metabolomics both in the peripheral blood and in the liver itself. And just with the infected samples, we observed two key signatures under these conditions. One was the presence of a dramatic increase in several species of corticoids, glucocorticoids, and the other important signature that we observed was a very large accumulation of a number of species of long-chain fatty acids in the liver. And we did validate the relevance of these signatures, because if we block the glucocorticoid receptor, the animals die much faster. And it's similarly, if we block CPT1 by this drug etomoxir, which is the first key step in the import of long-chain fatty acids into the mitochondria to use long-chain fatty acids, the animals also die considerably more and faster. So these signatures are actually relevant for the pathophysiology of this condition. You see here a drawing that explains how long-chain fatty acids are important into the mitochondria that we have seen to be accumulating under infection conditions. And by looking at the different components of this machinery, which are generally under the control of PPAR alpha, which regulates the expression of CPT1A, CPT2, and SLC25A20, which is this transporter that you see here, you can appreciate that all of them upon infection are substantially downregulated, and therefore this machinery does not seem to be fully operational under infection conditions. And you can see that substantiated here. We did this targeted signature of long-chain fatty acids. Indeed, infected samples show a prominent induction of all these species, while when they are treated with doxycycline, we see a substantial decrease in the accumulation of these species. And this is really relevant, because if we use doxycycline to treat these animals in the presence of the block of this pathway, then we can no longer improve the prognosis of these animals, which die just similarly. Now then we turn to the other signature, which is the accumulation of glucocorticoids. And here is perhaps a bit more complex, because with infection, as I demonstrated before, they're strongly upregulated. But even with the treatment of doxycycline, they don't normalize their presence. And so we reasoned that perhaps the glucocorticoid receptor would be responsible for this, and perhaps that's where we could see relevant differences. And sure enough, by looking at markers of the activation of glucocorticoid receptor, we could see that, as it was known, in the presence of the infection, the markers of activation are down, suggesting that the glucocorticoid receptor is inactivated, which is widely accepted as one key mechanisms of the severity in sepsis. And what we could then observe was that with the treatment with doxycycline, we could actually regain some of these markers of activation, suggesting that we could improve the sensitivity of the glucocorticoid receptors with this drug. And indeed, if we treat with a blocker of the glucocorticoid receptor in the presence of the drug no longer can act. So suggesting that indeed this is very important, the mechanism of protection that we are observing. So we try to bypass this problem by supplying medium or small chain fatty acids, which do not require this machinery for the import into the mitochondria. We could see some trends in improving the outcome, but they were not sufficient to really improve survival. Neither was an agonist of the glucocorticoid receptor. So we had to conclude that if we act on only one of the pathways, that would not be enough, and only the synergy of these two pathways was enough to improve the survival of these animals. So going further down into mechanism, one of the key questions would be how the doxycycline was acting, because it's an antibiotic. It could be acting through modifications of the microbiome at the level of the gut or at the level of the lung. These policies. Therefore, we use germ-free mice to test this hypothesis, and we could observe that even in germ-free mice, doxycycline could substantially improve the survival of this sepsis model. And so, while we do not exclude an effect at the level of the microbiota, it is also clear that the drug is acting through the host to protect in sepsis. So here is a structural view of the mitoribosome. You can appreciate that all of these classes of antibiotics have different according to that, they have slightly different mechanisms, but they all in general work by blocking a protein synthesis in a mitochondria. And you can see that tetracyclines bind to the smaller subunit of the mitoribosome, while chloramfenicol actually binds on the larger subunit. So therefore, they could have finer different mechanisms. So in general, and for a long time, tetracyclines are known to inhibit protein synthesis in a mitochondria by preventing the binding of amino acid transfer RNA to the A site. You probably know about this. And the other important information that I wanted to convey to you is that the assembly of the electron transport chain, which is critical for the function of the mitochondria, depends on components which are encoded at by the mitochondria, but most of the others are actually encoded by the nuclei. So the mitochondria genome encodes 2 ribosomal RNAs, 22 transfer RNAs, and 11 messenger RNAs. 2 of them are bicistronic. So overall 13 protein components, which then have to assemble in precise stoichiometries with the subunits which are encoded by the nuclei to produce functional complexes of the electron transport chain. So we have complex 1, 2, 3, 4, and 5. And with the exception of complex 2, which is exclusively nuclear encoded, all the others result for the assembly of components coming from both places. So it's important that these stoichiometries are precise, otherwise you will generate signals to indicate that there is a disruption in this balance. And you can see when animals, and then we sample the livers and look for mitochondrial components which are exclusively mitochondrial encoded, we see a decrease in their presence, while the ones are suggesting that there is an imbalance, and this imbalance can create the signal for compensation of this perturbation, and that's perhaps the source of the protection in these animals. So we have substantiated the outcomes in terms of what they do to the function of these different complexes. So a treatment with the doxycycline fundamentally affects complex 3 and complex 4. So to move on from a purely pharmacological way of demonstrating this, we then used a genetic tool to measure the potential impact of the perturbation the electron transport chain. And in order to do that, we use knockouts for CRIF, condition knockouts for CRIF1, which is an important component that is critical for the assembly of the ribosomal subunits that are nuclear encoded. And when we use these mice, we can see that the first complex to be affected in mitochondria is complex 4, which is an interesting parallel with what we observe for the treatment with doxycycline. Now, this is to show you the validation that at mRNA level, if we deliver CRE to the liver, which is has been done with adeno-associated virus 8, which is really very specific and effective. You can see almost complete depletion of the mRNA for CRIF1. At the protein level, we have depletions in the order of 30%, which gives us a partial depletion, which is just perfect for what we want to do. And in that case, if we actually use homozygous mice and we deplete CRIF1, we see that actually the mice do not have any survival advantages. But if we partially deplete with this genetic tool the presence of CRIF1, then we have a substantial protection. And similarly, there is no change in bacterial burden, as I've shown with the drugs. So this seems to mean that a partial perturbation of the mitochondrial function does have a protective role in this condition. So we ask whether this would be true only for complex 3 or 4, or what complex of the electron transport chain if we would still have protection. And therefore we use another drug, which is fenformin, and this is a highly potent and specific inhibitor of complex 1. And so if we do this, we actually have a very robust protection of the mice for this model. Thing is that we achieve this with a single application at the time of the procedure, because if we actually do 2 applications, the mice die actually faster. And this to us tells us that it's the partial and transient inhibition of the electron transport chain generating a signal for a compensatory mechanism that is driving protection. And again, with fenformin, this is disease tolerance because bacterial burdens are not affected. So we can validate this. Also with fenformin, we have a similar pattern to doxycycline. So we largely normalize, we dramatically decrease the accumulation of long-chain fatty acids. And actually, then if we do RNA-seq substantially later, so that we expect to find a transcriptional signature, indeed that is the case, we were able to find some key, candidate reasons or mediators for this protection. So we found, for example, adrenergic receptors to be strongly modulated by the presence of fenformin. So adrenergic receptor alpha and beta were changed, and in order to starting to distinguish which kind would be more important, we treat the mice with fenformin and with an alpha or a beta adrenergic receptor type of blocker. It's interesting because when we block also the alpha receptors, adrenergic receptors, we don't have we still have the same level of protection, but if we use propranolol, which is a general inhibitor of beta-adrenergic receptors, then we lose protection. And this seems to suggest that beta-adrenergic receptors are involved in the mechanism of protection induced by protein synthesis in the mitochondria, which we can really substantiate by looking at the lipid accumulations, both using histological methods, but also using mass spec. We can see dramatic differences with fenformin, which are affected by the presence of beta-adrenergic blockers. So in summary, it seems to suggest that protein synthesis inhibition in the mitochondria, if we do it in a transient manner, is highly protective against sepsis. And the key mechanism mechanisms for this to happen seem to depend on increasing sensitivity not only to the glucocorticoids which are activating during sepsis, but also to sensitizing the mice to the circulating catecholamines through the induction of higher levels of beta-adrenergic receptors in the liver. So this contributes to the idea that of core cellular functions, as I've shown you for DNA damage and now for inhibition of protein synthesis in the mitochondria, seem to drive a protection response, which I've shown you to be due to the induction of disease tolerance, but it's also possible that does it in another way. So just to put these things in context, and for those of you interested in immunology, you might be aware that there is now what I call a standard model to explain how an immune response is initiated, and this is the so-called pattern recognition receptor model, and this says that the effector cells, not only effector cells, but cells in the host, have pattern recognition receptors which can detect conserved signatures in different groups of pathogens, and that initiates signaling, telling the host that a foreigner microorganism is present. There is another very well-known model, which is called a danger model, which basically states that the host react not to the presence of a microorganism, but actually to the consequences of its presence. In other words, the damage or the disruption caused by that model, by that organism. And there is at least variations and a third model, which is called effector triggered immunity. And this is a model that states that the host reacts to virulence factors of microorganisms. And this is largely observed in plants. There's not so much data to state that this actually exists in other organisms, including vertebrates, as this would be nearly impossible for the host to be able to target all the variations of virulence factors. Certainly would not be compatible with including all these factors in the genome. So this seems to be operational in plants, but there's not a lot of evidence for organisms, and this seems always to be indirect. What we are now proposing is that perhaps there is another model that does not work by itself, but it's probably a required component effective initiation of an immune response, and this is to couple the detection of signatures of microorganisms, but actually to distinguish whether they are pathogenic or not, or pose a threat to the host, is actually to detect deviations in homeostasis caused by those organisms. And to this we call surveillance immunity. We have evidence that these perturbations may not be able only to induce disease tolerance, but also in initiating resistance mechanisms, as you see here, preliminary data by student in the lab, Katia Jesus, who has produced initial data to demonstrate that actually perturbations of these same core cellular functions can also clearance of the pathogen. So you can see that in early time points, specifically at 24 hours, as I demonstrated before, the bacterial burdens are exactly the same. Actually, the clearance of these bacterial burdens actually is faster, and this is compatible with the lateral activation of resistance mechanisms. This, I would say that the successful immune response depends not only on the direct detection of a microorganism, which is normally done in principle by the standard model of, also by the detection of perturbations in homeostasis induced by the presence of these organisms. And the conjugation of those leads to the induction of immune effectors and homeostasis factors, and this by this synergy that you can really induce survival of the host. So I mentioned that this is the work mostly of Enrique, who's no longer in the lab. with substantial contributions of more people in the lab, notably André and also Tiago, and I'm happy to answer any question you might have.
Thank you. Luís, does anyone have a question for Luís? No? So I have a question regarding your model of infection. So I assume that you are using an E. coli that is resistant to tetracycline.
Yes,
so
that is correct.
The resistant is through Tet-A, right? So it's
that's correct.
Yeah. So
it's a pump that puts out the drug.
So I was thinking about bacteria pathogens that actually have enzymes that can degrade tetracycline. Would you expect to still work in bacteria that can degrade tetracyclines, for example, Pseudomonas aeruginosa, or?
Yeah, that would be irrelevant for what we're saying here, because the idea here is that we are demonstrating that the drug also works in the host, which is not a big surprise, and it's actually known for several decades that there are certain classes of antibiotics that work much better than it would be anticipated by the elimination of the pathogen alone. And they are normally classified as immunomodulatory, and the mechanisms are not clear, but they just seem to resolve the infection and specifically inflammation much faster and much more effectively. And that is not explained by their direct effect on the bacteria that we are trying to eliminate, because if you do it in vitro, their effectiveness is just the same. But certain classes of antibiotics, like we're now showing for tetracyclines, but also for fluoroquinolones, for example, are known for a long time to do that. So if we think that we would be treating sepsis, we would not treat them alone with these antibiotics. This would be to target also additional mechanisms in the host and promoting these kind of mechanisms that induce disease tolerance and so preserve organ function, which is something that has not been done to treat sepsis. You treat infection, you sustain organ function in very unspecific ways, and you hope for the best. And what we're saying is that you have to act more proactively and induce those states by targeting the pathways that we begin to understand are critical to preserve organ function and to minimize tissue damage.